Nice introduction. I'd like to begin today first by thanking you for the opportunity to come and share with you what we're trying to build out on the Anschutz campus. To begin with, I'd like to give a little bit of background so everyone is in essence on the same page as we begin to go forward with the lecture. First, let me just discuss a little bit about the properties of both embryonic stem cells and adult stem cells. Embryonic stem cells, I think all of you have heard about because this tends to be what the media really wants to focus on. And certainly that has been the case for the last 10 years. The real excitement in the field initially concerning embryonic stem cells is the fact that these cells are actually pluripotent. I don't think that the pointer's going to work, so I'll just explain things as I go through. So the fact that they are pluripotent means that they can actually give rise to every cell type within the human body. So this was the real excitement about the potential of using these cells for regenerative medicine. I think, as all of you know, the real downside is the political and ethical issues of having to destroy an early fertilized embryo in order to harvest these cells. Adult stem cells, on the other hand, you probably have not heard as much about, but in fact, adult stem cells have been used clinically for over 40 years, and I'll show you some examples. When I came here in 2007 to begin the new stem cell program, we decided that we would only focus our efforts on adult stem cells. And hopefully by the time I finish with this lecture, you'll understand why. It was not only because of ethical and political issues. We used to think that adult stem cells were actually very limited in their ability to give rise to other tissues. For example, if you isolated a stem cell, let's say from the skin, we thought that those cells would only give rise to skin. A stem cell from muscle, only to muscle. But as I'll show you, this view has changed dramatically in the last 3 years. And in fact, I'll show you that we can actually now reprogram these cells back into a cell that looks very much like an embryonic stem cell. So first, I'd like to give you a few examples of current applications of adult stem cells. I think everyone in the room has probably heard of bone marrow transplants. Bone marrow transplants involve taking a small sample from the bone marrow. This sample contains hemopoietic stem cells. These give rise not only to the red blood cell lineage, but also to lymphocytes, which are important for mounting an immune response. So bone marrow transplants have actually been carried on routinely now in the clinic for over 40 years. So a very safe example of how adult stem cells can be used to regenerate not only blood, but also lymphocytes to fight infections. The next example is a little bit graphic, and I suspect that you haven't heard about it, but this is using skin stem cells to regenerate skin in patients who have severe burns. Again, this is a technology that's been used clinically for over 30 years, but I dare say most of you probably have never heard about it. So this was a procedure developed by Howard Green when he was at MIT. Howard discovered that you could take a biopsy from a patient that was burned in an area that had been spared from the burn, about the size of a postage stamp. You could put those cells in culture, and you can expand them over a period of about 3 weeks into enough cells to completely cover the entire surface of the human body. The first patients that Howard treated are still alive. That was 30 years ago. So what that says is, in that tiny biopsy, there are adult stem cells. When you put them in culture, you can expand those cells, and once you transplant them back onto the patient, they remember how to regenerate the skin over and over and over for at least now over 30 years. The real problem with these patients is how do we keep them alive during that 3-week period when we're expanding their cells. And in the upper right-hand panel, what you see is what's typically done is the patients are actually, the wound is, the burned area is cleaned, and then it's surrounded by either cadaver skin or even pig skin, because pig skin is actually very similar to human. What you want to do is keep the patient alive from losing water, from getting infections during the 3-week period when you expand the cells. In the second panel down on the left, you'll see a fibrin matrices. It looks a lot like orange jello. This is actually biodegradable, so the stem cells are actually grown on the surface. So if you look now to the middle panel on the right, you'll see that these cells then are transferred to the burned patient very much in a quilt-like fashion to cover all of the burned areas. So down on the bottom panel, especially over on the right, you'll see the patients. Cosmetically, this is not beautiful skin, but the patients live. So it really has restored their ability to retain and prevent water loss and to prevent them from getting infections. The next example, I think, is the most certainly graphic, and one that has only been used clinically now for about 10 years. This is a procedure developed by a colleague, Michele De Luca, who is in Modena, Italy. In the upper left-hand panel, you'll see an eye that was subjected to a chemical spill. So you can see that the cornea is completely opaque. There's a complete loss of vision. Michele and his colleagues discovered that there is a region between the cornea and the conjunctiva called the limbus, and this is where corneal stem cells reside. He discovered that he could take a tiny biopsy from that region, put those cells in culture, and expand the corneal stem cells, and then return them to the patient as an autograft. So in fact, if you look at the eye on the upper right-hand panel, that's the same eye months later after completely regenerating a new corneal epithelium. Just a wonderful example of using adult stem cells for regenerative medicine. So in addition to chemical spills, now so many people wear contacts. Sometimes if you get an infection, you can actually cause a complete destruction and depletion of the cornea stem cells. So there really is a very unmet need for developing this technology. I'll just show you in the next slide a wonderful example, I think is my most favorite example from Michele. This individual actually had chemical spills in both eyes. He had not been able to see in 20 years. But when he came into the clinic, Michele noticed, if you look in the upper left-hand panel, that there was a region of the cornea that was not subjected to the chemical spill. He biopsied that region and found out that the corneal stem cells were actually fine. It's just that the connective tissue where the burn from the chemical spill is not a good substrate for cells to migrate back in and heal the damaged corneal epithelium. So he put the cells in culture. He could expand them into enough cells that, in fact, he repaired both eyes in the lower panels. So this individual who had not been able to drive, had not been able to work in over 20 years, walked out of the clinic and went back to work and went back to driving. Just a wonderful example of using adult stem cells for regenerative medicine. So these are just to show you that this really is a permanent corrective therapy. On the left panel, you can see the clinical condition of the eyes of individual patients before they were repaired. And on the right panel, you can see follow-up. There's a follow-up of 2 years, 4 years, and the bottom right-hand panel, 5 years. So assuming these patients don't get another injury, if they don't get an infection, this is permanent regenerative medicine for regenerating new corneal epithelium. So Michele is actually an old colleague of mine. We both trained together at the NIH in the mid-'80s. He's actually visited Denver twice. Currently, there is not a cornea regeneration program in the U.S. There are actually patients in the U.S. who actually go to Italy to have this procedure done. Michele is very interested in helping us establish this program, a cornea regeneration program in Denver. We were in discussions with the chancellor, the dean, the chair of ophthalmology here, Naresh Mandava. We were well on our way to building a new building that would have a GMP facility. That GMP stands for Good Manufacturing Practices facility. This is a very clean facility where you can expand cells without a concern about getting an infection.